Overview of the cell cycle. The cell cycle and cell division are fundamental biological processes that ensure the proper replication and distribution of genetic material to daughter cells. Understanding these processes is essential for grasping how organisms grow, develop, and maintain tissue homeostasis, as well as the implications for cancer biology. Below is a detailed overview of the cell cycle, the mechanisms underlying cell division, and their regulation. The cell cycle is a series of phases that a cell goes through to grow, replicate its DNA, and divide. The cycle is typically divided into two main parts: interphase and mitotic phase, M phase. Interphase. The period of growth and DNA replication that occurs before mitosis. Interphase is subdivided into three phases. G1 phase, gap 1. The cell grows, synthesizes proteins and organelles, and prepares for DNA replication. This phase is characterized by active metabolism. S phase, synthesis. DNA replication occurs, resulting in the duplication of chromosomes. Each chromosome now consists of two sister chromatids. G2 phase, gap 2. The cell continues to grow and prepares for mitosis. During this phase, cellular structures required for division are assembled. Mitosis, M phase. The process of cell division, where the replicated chromosomes are equally distributed to two daughter cells. Mitosis is divided into several stages. Prophase, chromatin condenses into identifiable chromosomes, each consisting of two sister chromatids. The mitotic spindle begins to form, and the nuclear envelope starts to disintegrate. Metaphase, the chromosomes align at the metaphase plate, the equatorial plane, of the cell. Spindle fibers attach to the centromeres of the chromosomes. Anaphase, the sister chromatids are pulled apart and moved towards opposite poles of the cell as the spindle fibers shorten. Telophase, the chromatids reach the poles, and decondensation of the chromosomes occurs. The nuclear envelope reforms around each set of chromosomes, resulting in two nuclei within the cell. Cytokinesis, this process follows mitosis, where the cytoplasm divides, resulting in two separate daughter cells. Detailed phases of the cell cycle. Interphase. G1 phase, gap 1. Cell growth occurs, with an increase in size and mass. Synthesis of proteins, lipids, and organelles essential for cell function. Cells perform their specific functions and may carry out specialized activities depending on their type, e.g., muscle cells contract, neurons transmit signals. Restriction point, are a crucial checkpoint at the end of G1 ensures the cell is ready to enter the S phase. Cells can proceed to S phase, enter a quiescent state, G0, or undergo apoptosis. S phase, synthesis. DNA replication occurs, resulting in the duplication of each chromosome. DNA is synthesized via semi-conservative replication, where each new DNA double helix consists of one old strand and one newly synthesized strand. The entire length of the genome is replicated, ensuring that each daughter cell will receive an exact copy of the genetic material. G2 phase, gap 2. Further cell growth and preparation for mitosis occur. Synthesis of proteins necessary for mitosis, including tubulin for spindle formation. Organelles may duplicate, and the cell ensures all genetic material has been correctly replicated without damage. The cell checks for DNA errors and damage, engaging in repair mechanisms if necessary. M phase, mitosis. Prophase. Chromatin condensation occurs, forming visible chromosomes. The mitotic spindle develops from centrosomes, which are specialized microtubule organizing centers. The nuclear envelope begins to break down. Metaphase. Chromosomes align along the metaphase plate due to the action of spindle fibers. Each sister chromatid is attached to microtubules from opposite poles of the spindle apparatus, ensuring equitable distribution during division. Anaphase. The cohesin proteins binding sister chromatids together are cleaved, allowing them to separate. The sister chromatids move toward opposite poles, ensuring each daughter cell will have an identical set of chromosomes. telophase. Chromatids reach opposite poles, and the nuclear envelope reforms around each set of chromosomes. Chromosomes begin to decondense back into chromatin. The mitotic spindle disassembles. Cytokinesis, often considered part of M phase. The cytoplasm divides through the formation of a contractile ring, in animal cells, or cell plate, in plant cells. This results in the physical separation of the two daughter cells, each containing its own nucleus and the same chromosome number as the original cell. Cell cycle regulation. The cell cycle is tightly regulated by a network of proteins known as cell cycle regulators, including cyclins, cyclin-dependent kinases, CDKs, and checkpoint proteins. Key regulatory mechanisms include cyclins and cyclin-dependent kinases, CDKs. Cyclins, these are proteins whose levels fluctuate throughout the cell cycle. They activate CDKs by binding to them, forming cyclin-CDK complexes that drive the cell cycle forward. CDKs, these are kinases that, when activated by cyclins, phosphorylate target proteins to regulate various phases of the cell cycle. For example, CDK1/cyclin B is crucial for the transition from G2 to M phase. Cyclin-dependent kinase inhibitors, CKIs proteins like P21 and P16 inhibit CDK activity, serving as checkpoints to prevent the cell from progressing to the next stage until certain conditions are met. Checkpoints in the cell cycle. Cellular checkpoints are critical regulatory points that monitor and control cell cycle progression. G1 checkpoint determines whether the cell is ready to enter S phase based on size, nutrient availability, DNA integrity, and growth signals. Cells that do not meet criteria may enter a quiescent state, G0, or undergo apoptosis. G2 checkpoint assesses DNA replication completeness and checks for any DNA damage before the cell proceeds to mitosis. M checkpoint, spindle checkpoint, ensures all chromosomes are properly aligned and attached to the spindle apparatus before anaphase begins, preventing unequal distribution of chromosomes. Mitosis vs. Meiosis. While mitosis is the process of nuclear division that produces two genetically identical daughter cells, meiosis is a specialized type of cell division that results in four genetically unique gametes, sperm and eggs. Key differences and aspects of meiosis include meiosis I, homologous chromosomes pair up, synapsis, and undergo crossing over, exchanging genetic material, which increases genetic diversity. The homologous pairs are then separated into two daughter cells. Meiosis II, similar to mitosis, the sister chromatids of each chromosome are separated into four haploid gametes. Genetic diversity. Meiosis introduces genetic variation through independent assortment of chromosomes and recombination, crossing over, which is essential for evolution and adaptation. Implications for health and disease. The regulation of the cell cycle is crucial for maintaining tissue homeostasis. Disruptions in cell cycle regulation can lead to uncontrolled cell proliferation and cancer. Key points include cancer mechanisms, mutations in genes that encode cyclins, CDKs, checkpoint proteins, or tumor suppressors, e.g., p53, can result in aberrant cell cycle progression, leading to tumorigenesis. Chemotherapy. Many cancer treatments target rapidly dividing cells by interfering with DNA synthesis or mitotic processes, making an understanding of the cell cycle crucial in cancer treatment. Stem cells and regeneration. Cell cycle regulation is also vital in stem cells, which must balance self-renewal and differentiation, impacting tissue regeneration and repair processes.